This is CGTN, China Global Television Network.
This week on Razor, measuring intelligence.
Corvids use these intricate techniques or tactics of deception, which resemble some of the tactics that we do.
So they're doing magic with their mates?
They do a slide of beak, if you will.
And manufacturing mini human hearts.
Within days, we can mass produce and generate billions and billions of cells. So you can see these heart cells as the fundamental building blocks. And they become a mini heart that is fully capable of ejecting fluid.
It used to be an insult to call somebody a bird brain, meaning they didn't have much intelligence. But work here at the Cambridge Animal Behaviour Lab is showing that birds like these, that belong to the crow family, or corvids, are very intelligent indeed. And it's also shining a light on how intelligence itself evolved. Professor Nicola Clayton from Cambridge University is an expert on comparative intelligence. So, Nikki, which came first, the interest in intelligence or the interest in birds?
Oh, the interest in birds.
Really?
I've always wanted to fly like a bird, and I've always wondered what it's like to think like a bird.
What birds are we talking about?
The ones that I specialise in are members of the crow family, the corvids.
Right.
And corvids are extremely intelligent. They're feathered apes when it comes to their cognitive abilities, their ability to think about the here and now, to reminisce about the past.
You were very careful in our briefing for me to make sure I had had a shave, that I didn't have a white beard. And sadly, I do have a white beard. And why is that important?
Well, it's the Eurasian jays. Right. They're a bit fussy.
The ones we're going to meet this afternoon?
The ones you'll meet this afternoon. Not all corvids are this fussy, but the Eurasian jays don't like white beards because the white's in the wrong place. The white should be on your bottom.
Oh, right.
Not your chin. It's in the wrong place. Be a bit like, can you imagine talking to me if my face was where you expected my bottom to be? I think you'd be a bit freaked.
It would be a bit difficult, yeah. Eurasian jays, or simply jays, are small, colourful corvids found from Western Europe to Asia and North Africa. PhD student Eli Garcia from Cambridge University's Zoology Department is studying them. In specially designed booths where the birds can come and go freely, he shows them magic tricks.
Here we are.
So this is literally where the magic happens?
Literally, yeah. All right, so if you go in, you'll get a bird's eye view.
Oh, literally, once again, literally.
Yeah, literally. I would say the most interesting part of what we're doing in terms of magic effects is on the comparisons, right? Because remember, what we want to know is whether non-human animals, be that jays, be that chimpanzees, be that whatever, have the same expectations as humans.
Why do you use magic?
Well, magic capitalizes on your expectations, right? If I grab this coin, you have an expectation of what I just done, because that's what normally happens when somebody grabs a coin.
I expect it's in that hand.
You expect that, yes,
Yeah.
but that's the beauty of magic, right? It works on surprising your expectations, right?
That's cool. That was cool. That's very cool.
The interesting thing here is, why do humans develop these expectations, and do non-human animals also have them? Do non-human animals think about the world in the same way that we think about the world?
Right.
Okay, so you see the coin.
Yes.
I grab the coin.
Yes.
Yeah.
So I expect that you've grabbed it and it's in that hand.
But like, you stopped seeing the coin very early on in the effect, right? Yeah. Everything that happened in between, you thinking where the coin is, you envisioning, you imagining where the coin is. You did that. I didn't do anything.
Exactly.
Right?
Yeah.
I just showed you a coin and then hit it.
And you dropped it in your other hand.
Exactly.
Right.
So if you break these in the cognitive mechanisms that you need, right, the mental mechanisms that you need in order for you to understand these, the first thing that comes to mind is called object permanence. So even if you cannot see, hear, taste, or smell it, the object still exists.
And the evolution of object permanence is vital to a particular survival strategy.
So corvids do something very interesting. They hoard, okay?
Yes.
They are hoarders. They just grab as much food as they can and they save it for later consumption.
Right.
But what they do is that they hide little bits of food in the soil.
Yeah.
And that's called a cache.
Right.
And the behavior is called caching. The interesting thing is that object permanence, so this ability to actually understand that things, even if hidden, even if not observable, still exist, develops at the same time that they learn to hide things.
And this comes back to what Nikki told us about this morning, because it's also about this idea that they understand there's a future, because they're putting that food away for a time, for the future, when
Absolutely.
they don't have food.
Yeah,
maybe don't have food.
Yeah. I mean, the theory of memory is that memory evolved with the future in mind, right? We remember things because we might need them in the future.
Oh, that's really interesting. So it's not about the past at all, it's about the future. So that describes evolutionarily why we have memory.
Yeah.
Because we can remember where we put that antelope we caught.
Yeah, absolutely. That's actually the best description, I would say. So we can remember where we put the antelope.
I hate this question, but as science journalists, we sort of feel obliged to ask it. Why do we need to know this? Why do we need to know how brainy birds are?
Well, you might be curious. If you're a bird nerd like me, then it might be very important to know it just out of curiosity. If you're only really interested in how humans think, then you want to know whether other animals think in the same way as humans or not for comparison. So the more distantly related the animals are, the more it tells you about the whole idea of whether you've got convergent evolution happening. In other words, intelligence evolving independently in very distantly related animals and understanding why that might be.
Oh, so that's really interesting. You're wondering whether intelligence evolved several different times for
Yes.
different reasons?
For different reasons or for the same reasons. You know, we shared a common ancestor with birds over 300 million years ago, and we have mouths and they have beaks, we have arms and they have wings. There's many other differences, of course. So of course there are differences, but looking at the pattern of differences embedded within similarities. So to me, the fact that these corvids, they can imagine the future and think about the past is fascinating.
Using magic as an experimental technique to help answer these questions is particularly relevant for the corvids, because in the wild, as part of hoarding their own food, they use a little magic themselves.
So corvids use these intricate techniques or tactics of deception, which resemble some of the tactics that we do, in order to confuse the onlooker into thinking that they've cached somewhere else. So the onlooker, the pilferer, doesn't steal their cache.
So they're doing magic with their mates.
They do a sleight of beak, if you will.
Hi, girlie. Come get. There you go. Good work. It's time now for Eli to demonstrate the magic he's been using with the Eurasian jays.
Satsuki. Come on, Satsuki.
The birds have not been working, as Eli calls it, since the beginning of the pandemic, so they're a bit shy. Is that a he or a she?
I'll tell you once I see.
Here she comes. Yeah, she's having a good look at the camera.
Come on.
Come on, baby.
You see, she's not convinced by the idea of... She'll come in, have a warm and then leave again, most likely. But... All right, let's see if she remembers. So I'm just gonna... Babe.
Sadly, Eli is not getting willing assistance today because the birds are out of practice. but he can show us some of the work he's submitted in his recent paper.
So this is the palm effect.
Yeah.
I fake a palm, but they don't care that I faked it.
She gets it.
She gets it. Yeah.
But you fooled me.
Yeah.
Which
and
is
now I
embarrassing.
do it slowly, and then
Oh, that's so
she gets
beautiful.
it, and then I do a real transfer, and she doesn't get it.
The birds get this trick right about as much as they get it wrong, which tells Eli that the movement does not offer them enough observable cues to make a clear choice. However, a human is fooled by this trick because they expect the worm to move from one hand to the other.
So this is the French drop for you.
This is the one that even though I knew what you were doing,
Yeah, exactly.
I still couldn't see it happen.
That's my hand.
Yeah.
And then I'm grabbing it.
Yeah.
And then the bird is choosing. And you can see he's choosing correctly, so the hand that I started.
So you're not fooling the bird very much.
Not with these two effects, no, because
Right.
they work because you have an expectation on hand movements.
Right.
Why should a bird have an expectation of hand movements?
Oh, because the bird's got wings.
Exactly.
Oh, that's a relief. I thought I was dumber than a bird.
No, birds got wings. On that point,
Yeah.
let's have a look at the magic effect
Right.
that actually capitalizes on something else,
Yeah.
such as your failure to see fast movements.
Right.
You'll see that the bird actually gets totally fooled by these. So
What's this one called?
The fast pass. So
Ah,
it's the
right,
one-
right.
So if I do the fast pass, they see it in the right hand. Then I do the movement, and they still think that it's in the right hand,
Right.
even though I moved it very quickly.
But they've got better frame rate than I have, though. haven't they?
Exactly. That's the astonishing thing, right?
Yeah. Despite being able to see more frames per second than humans, the birds still miss the trick because their eyes are on the side of their heads. That means they have the choice of using only one eye, monocular vision, or using both eyes, binocular vision.
So basically, this magic effect capitalizes on the fact that she's switching from monocular vision to binocular vision. And in the switch, in between monocular and binocular, there's a blind spot. So she misses where the magic happens. She cannot see it.
But what does that tell you about intelligence if it's actually about a blind spot?
It tells me about perception,
Ah,
not about intelligence.
Right.
But do bear in mind that intelligence starts at perception, because we perceive the world around us, and then we think about what we've seen and what we have perceived.
right. We input the data and we analyse it.
Absolutely.
It's magic. Well, it is magic. Myth and literature, we've got lots of examples of brainy corvids, of intelligent corvids, crows, ravens, rooks. Didn't we already know this stuff?
Well, it depends what you mean by know, right? So we had lots of general artistic knowledge suggesting that this was the case, but we didn't have scientific evidence to support that knowledge.
So what you're doing is you're taking folk knowledge and turning it into scientific knowledge so that we know they're bright, but now we understand why they're bright.
That's right. By developing experiments to test some of these hypotheses or questions and finding out whether the answers to our questions are just assumptions or whether there's actually proper evidence to substantiate those claims.
And these studies also remind us that what we call differences in intelligence are really often just about the different ways we perceive our world. A lesson as relevant for our understanding of ourselves as it is for our understanding of other species.
You can see more of Razor on our YouTube channel. Search for Razor Science Show and it will take you straight there. Remember to subscribe and hit the bell button for notifications.
Facing the unknown is always difficult. In a world in turmoil, it's easy to lose orientation. But when the storms come, we have to see the possibilities, reinvent, find new opportunities, discover a path forward. CGTN. See the difference.
On the agenda with me, Stephen Cole, we look up into space. We look down into data. We look at debt. We look at politics. We look at opioids, climate change. We look at all the issues that really matter around the world. But you matter too. We want to tell the stories you want to see and hear about. Make it your agenda.
How will your world change today? What happens here? What happens there? Or what you make happen for yourself? If it matters to you, it matters to us too. Your stories are the stories that need to be told. Africa Live. Find your voice.
Ever wondered what's the difference between a bear and a bull market? Where are the cash cows? And who are the lame ducks? And what exactly are black swans? Grey rhinos? And unicorn companies? Make sense of it all with Global Business, only on CGTN.
Globally, cardiovascular diseases are the leading cause of death. The World Health Organization says that in 2019, an estimated 17.9 million people died from these diseases. That's 32% of all deaths. The majority were due to heart attack and stroke. And as people are now living longer, often with multiple diseases, this will become more common.
There are so many major hurdles in cardiobiology. For instance, the heart doesn't regenerate. So the heart is damaged, the damage is there forever. The heart cells are gone, they are gone forever.
There are therapies for heart disease, but how do doctors know which ones work, and how can new drugs be tested safely and effectively? That's where biomedical company Novoheart comes in. Owned by the Madeira Group for Drug Discovery, it's come up with a revolutionary solution bioengineered from real heart tissue.
And here it is, a so-called heart in a jar. A mini living human heart pump, the first of its kind. They're grown from pluripotent stem cells, which can also be turned into other types of cells and tissues in the body. And they could be a game changer, helping to streamline and reduce the costs of testing medicines for heart disease. Professor Ronald Lee is the CEO and founder of Madeira.
Whenever a drug company wants to develop a new drug, so be prepared to spend about 10 to 12 years of time, several, let's say 2 to 4 billion US dollars, and yet the chance of being successful is less than 10%, or even some people would tell you even less than 5%. And the main reason is that everybody has been really using animals like rats, mice, you name it, for screening. And also whenever a drug fails, 50% of the chance, it has to do with side effects of the human body.
Cardiac toxicity is a common serious effect of numerous new as well as marketed drugs, not just those for the heart. But Madeira's Novoheart is able to monitor the effectiveness and safety of drugs on several mini hearts in the lab at once, saving time. It also means no testing on animals or the patients themselves. All they need to create these heart constructs is a single drop of blood from a donor.
How do you grow these hearts?
So we have figured out a way to instruct the stem cell to become human ventricular cardiomyocytes, or human heart muscle cells. So within days, we can mass produce and generate billions and billions of cells. So you can see these heart cells as the fundamental building blocks. So once you have the building blocks, you can start assembling different tissues as well as a mini heart. And we do so by putting the cells into a specially designed bioreactor. So you can think of it as some kind of 3D printing, with the scaffolding, the extracellular matrices that the biologists call it. So allow a few days for the heart to grow and for the cells to come together, and they become a mini heart that is fully capable of ejecting fluid.
Heart muscle cells, or cardiomyocytes, are unique because, unlike other muscle cells, they're highly resistant to fatigue, constantly contracting and relaxing, which controls the rhythmic beating of the heart. It takes 10 million heart muscle cells to manufacture a mini heart, which is just the left ventricle, one of the 4 chambers of the heart. A metal shaft acts like the aorta, the main blood vessel. When the mini heart pumps, it generates pressure and ejects fluid out of the metal shaft, which means researchers can measure pressure and look at the mechanical properties of the hearts. With this technology, they can make healthy hearts for screening toxicity as well as sick hearts for efficacy.
The reason why we're interested in the left ventricle is because it's basically the engine of the heart, and diseases that we're interested in, like myocardial infarction, heart attack, heart failure, they're basically left ventricular diseases.
Heart muscle cells have many applications. A cardiac tissue strip, like a fibre, can be manufactured with 1 million cells. And Novoheart is also developing heart patches to repair damaged cells.
But there's only a certain limit by which you can shrink your heart down to a critical size when you can still successfully model many of the human physiological parameters.
Nailing the miniaturization aspect of the lab hearts proved to be a very key turning point and came out of a collaboration with fellow University of California scientist Michelle Kine. Razor featured the biomedical engineer's creation inspired by Shrinky Dinks, the flexible plastic sheets that shrink dramatically when heated but maintain shape and colour. Dr. Kine used the sheets to make microfluidic devices which can help diagnose medical problems using body fluids.
So we can pattern very inexpensively because we use Shrinky Dinks, which was a really popular children's toy when I was a kid. And so we pattern different materials onto the Shrinky Dinks. And if you remember back to those fun children's toys, you just pop them in an oven for 3 minutes and they come out. And instead of using them to make little jewelries or Christmas decorations, we actually take the active layer off. So we take the thin layer of metal or nanomaterials off and we put them into a soft stretchy material to allow us to get very high resolution monitoring.
So one very special features for the heart to function properly is the asymmetry. So for example, the electricity in the heart has to follow certain pathways in order for the heart to be able to properly contract. So that has to do with the symmetry. And that asymmetry is the result of cardiomyocytes or heart cells being aligned very systematically. And it turns out that her Shrinky Dink inspired technology allows us to be able to align cardiomyocytes or human heart cells in some very pre-designed systematic fashion. So that led to an engineered tissue design and also a series of studies, and with this tool, a very interesting tool.
For the first time, Professor Lee and his colleagues were able to see for themselves what effects several drugs had on a heart, albeit a mini one. In the past, antiarrhythmic drugs were mainly so-called blockbuster drugs, a one-size-fits-all drug earning a revenue of over $1 billion. With mini hearts, Novoheart is able to personalise antiarrhythmic therapy, guiding the development of drugs targeting specific problems. They can also develop hearts that carry human disease mutations and then trial therapeutics to return normal function. Dr. Roger Hajar is Madeira's Chief Medical Officer and CEO of Sadacor, a solely owned subsidiary of Madeira responsible for clinical trials. He's involved in the execution of clinical trials by choosing the most suitable therapeutics discovered using the mini heart and other platforms.
So you're not just treating disease before it becomes life-threatening, are you?
So we have multiple different platforms for treatments. Some, as you mentioned, are early on so that they don't progress to what we call end stage. But as we know, there are many patients at the end stage level from a cardiovascular perspective who need to have very specific treatment. So we also address that need, and it's a big unmet need. And gene therapy really comes into play in those settings where you have an end-stage disease that you're trying to reverse back, and you need to reverse it at the molecular level.
In rare or inherited diseases, gene therapy has been used for many years. The therapy targets specific abnormalities, replacing the missing or defective gene in a diseased organ. To deliver gene therapy into the heart, a catheter is inserted through the arm or groin. The therapy product, in green, is carried to the heart by an inactivated virus, which means it cannot replicate, and is injected into the coronary arteries. From there, these vectors carrying the genes are then distributed throughout the heart muscle, and they go into the heart cells and deliver their payload. That therapeutic payload is then distributed across the cardiac cell and translated into proteins. The proteins attach to the organelles or specialized structures that serve specific functions to keep a cell alive. So they improve the function of the cell and also the whole heart, turning a failing heart into a normal heart. Dr. Hajjar says that so far there have been no safety issues with the delivery protocol for human patients. Now Madeira is focused on efficacy in multiple heart failure settings.
In another case, we can basically, instead of genetic mutations, we can basically train these hearts in a jar to become like what we see in humans, a very specific disease called heart failure with preserved ejection fraction, meaning they're very thick hearts that can't relax. And we can basically make these hearts in a jar to mimic that disease. And then we deploy our therapeutic and test and see, well, is it improving? And that would give us a direct view of what would happen in humans, because these are human hearts in a jar, and they're not experimental models in animals, which sometimes do not directly correlate with what happens in humans.
Madeira has used healthy and sick mini hearts to discover new drugs and gene therapies, and has US FDA approval to start a clinical trial. A number of other preclinical candidates are being fine-tuned to advance to the clinical stage. Madeira is also developing smaller sized tissues to increase the speed and efficiency of screening these clinical candidates.
Is this the future? Will this completely change healthcare?
For us, we've been working on the mini hearts, and my other colleagues are developing mini brains, mini liver, mini pancreas, blood vessels, you name it, right? So basically, by combining all these mini organs, we can make mini me, but except it's not like a walking individual, but it carries all the major critical organs. So human patients are not the guinea pigs anymore. And you can kind of like think about it as some kind of blood test. You go into the clinic, take a single drop of blood or some sort of tubes, and for testing this and that. And on top of that, the blood can be used for deriving these mini organs for testing a number of different things, and even the future of medicine. It's very exciting.
With the vehicles charged, it's time for the...
What is going to happen now? Excuse me. Do you mind if I ask a quick question? Do you know what these cars are going to do now?
They're going to go shakedown.
Straight down where?
Shakedown.
Are they going to do shakedown? What does that mean?
Wow, you guys work here?
The shakedown, it turns out, is a practice lap. The 28-year-old Christine Giampaoli Zonca will be taking part.
Which is your vehicle?
1, 3, 4, the fourth, starting from the right.
With the purple and green. Look, were there points in the tour where you looked at some of the tracks and thought, this is going to be a real challenge for the vehicle?
Oh yeah, every time.
Really?
In this championship, every time.